Hello, everyone, and thank you for listening to my submission. Today, I'll be talking about my thesis topic, analyzing ligand specificity to assess the evolution of TLR4. Why this topic is important to look at lies in the definition of what TLR4 is. TLR4 is a protein which activates inflammation in response to bacterial infections. For instance, let's say you fall down and get a scrape on your knee, which bacteria enters. Your body is able to recognize that bacteria through recognition of LPS, a component of gram-negative bacteria's cell membrane, which is unique to each bacteria and can even vary among different strains. TLR4 dimerizes in response to this bound LPS, activating inflammation, which increases blood flow to the afflicted region, helping your body fight off the infection. However, when inflammation activates excessively, it can negatively impact your health, leading to arthritis, neurodegeneration, and sepsis, which contributes to 11 million deaths a year. As overactivation of the inflammatory pathway is deadly, the Harms Lab aims to study TLR4 rules for dimerization via evolution, as the stepwise changes allow us to observe alterations in structure and function to observe the subsequent effects on what they bind to, otherwise known as ligand specificity. As we can see in the graph, our lab has previously been able to look at various mammals' response to LPS. This has allowed us to look at which amino acids may have an effect on what different TLR4s bind to. However, as we start testing species that are further away from humans in our model system, which is human embryonic kidney cells, we see a major decrease in observable activity. This decrease is represented by Xenopus. However, other species of interest have also shown this response, such as pike. As our lab wants to be able to observe the activity of these low-signal TLR4s, we have come up with a theory and subsequent method of why these proteins are not activating and how we might increase that signal. Our theory is that species-specific ectodomains, which bind the LPS, can be put on human TLR4 to increase signal. And that theory is shown in the bottom right diagram. What we think is going on is shown by the upper right diagram, where basically these human TLR4 have these square inner signaling proteins, which are able to activate inflammation. However, when frog TLR4 are put in this human background, the human embryonic kidney cells, they're having to actually interact with these square inner signaling proteins when they need a circular one. So they aren't able to transduce the signal even when they are dimerizing. And therefore, we are unable to observe activation. Thank you for listening to my submission. I hope you have a wonderful day.